Hi guys, it's Mr. Y. Today we'll be continuing our genetics unit with the discussion over cell division by the process of mitosis and by a new process called meiosis as well, and how the terms chromosomes and genes are integrated into this, as well as covering how DNA itself replicates itself for both of these processes. So recall that the term chromosomes refers to DNA in a very compact and mobile form, and that we don't see chromosomes in the nucleus of most cells because during the life cycle of most cells are in what we call interphase. And so during interphase, the DNA is uncoiled in this form we call chromatin, so the cell just looks like this dark spot. But when the cell is getting ready to divide, that's when the DNA will coil up and become chromosomes, especially in what we call eukaryotic cells, cells that have a true nucleus, like plant cells, animal cells, protocells, and fungal cells. They're just very compacted forms of DNA, and again, they only appear just before the cell is about to divide. I'm pretty sure I've shown you this slide before. The classic shape for chromosomes is this X structure, and the center area is called a centromere, and then you have these four arm sections. These are, one section is called a chromatid, and then another section is called its sister chromatid. And again, typically this looks like an X shape, but you won't always see it in this X-like format. Remember that though the chromosomes actually make the DNA visible to the microscope, because as DNA is uncoiled most of the time, the DNA itself here on the left is actually too small to be seen even under microscopes. It's only when it's been coiled up again and again around these histones again and again and again to make it thicker that it becomes wide enough, so you can see it goes from 2 nanometers to 1400 nanometers to actually be seen via microscope. So again, you will see the chromosomes, which is the DNA, coiled up, but only before the cell is about to divide. And again, they're held together, these chromatids, each arm here is held together by the centromere, and you can see that some of these arms are actually longer than others, and some of these are shorter. This is actually what they look like under electromagnetic microscope. Excuse me. Again, centromere would be right about here, and again, you can see there's 4 different arms here. Again, that's just coiled DNA, and when it's uncoiled, it's really hard to see it under your typical microscope. But again, you only see the chromosomes just before the cell divides. So this picture is nice of the chromosome, I should say. It's not really accurate for the cell itself, because again, if this was truly when you'd see the chromosomes, the nuclear membrane here would be dissolving, because this would be prophase, for instance, of mitosis. Most of the time you just see this dark spot where the chromatin, the uncoiled DNA, sits. But when it is coiled into these chromosomes, they actually do have names for these different arms. There's what's called, the shorter arm are called the P arms, and then the longer arms are called the Q arms. And ideally, one of these chromatids should be an exact copy of the other. Not a mirror image copy, but an exact copy. So all the A's, T's, G's, and C's that are here should match all the A's, T's, C's, and G's that are here. Base pair by base pair, assuming everything was copied properly. And again, this is where you get that phrase sister chromatids. So this is one chromatid, this is another chromatid. They should be the same. That's why they're referred to as sister chromatids. The ends of these arms up here and down here at the end are called telomeres. Those telomeres actually kind of act like a bit of insulation as you copy the DNA. And they're very interesting in that there's a relationship for how many times a DNA chromosome has been copied and how long those telomeres are. It's thought that the telomeres generally get shorter and shorter each time a chromosome is copied, which is thought to kind of put a maximum age limit over most organisms, because as the telomeres wear off, as they get worn down, you get to genes that are actually really important for the organism to survive. The telomeres themselves, they don't code for any proteins. They don't actually do anything except act kind of as this insulation. But once they're worn away and you get to the coding DNA, the DNA that will code for proteins, and that starts to wear away, then you have cells that won't survive. So it's thought that the length of the telomere is actually associated to how long organisms can survive. For humans, it's estimated that our maximum lifespan right now, the current estimates put it at about 122 years, give or take, which is about, the current record, I think, is somewhere around 118, 119 years old for the oldest human ever. And it's thought that you can't go beyond that without somehow figuring out a way to maintain the telomere length or stop it from, or increase it again. And again, different organisms will have different lengths to the telomeres. Some will wear out faster than others. And as the telomeres wear out, you also see a higher increase for rates of cancer. So older people tend to have higher risks of cancers, and part of the reasoning they theorize, is because telomeres on their DNA are wearing out, and so you're getting closer and closer to the actual important DNA on each of these arms that's more towards this center area, the DNA that actually codes for important proteins. And as that DNA gets worn down, then cells don't survive, they become mutated, and you can have things like cancer crop up. So this slide is a reminder of the fact that there's two variants of chromosomes. There are what are called single chromosomes and then what are called double chromosomes, or sometimes replicated chromosomes. Both technically get the term chromosomes. They're both held together by a centromere, but you can see one has 2 arms and the other has 4 arms. Now, typically, the double chromosome will show up just before the cell is about to divide, because you need to have double the DNA. So there's the original copy of the DNA, and then here's the replicated copy of the DNA here in the red. So these two sides should have the exact same base pairs. All the A's, T's, C's, and G's should match up the exact same way. Then when the mitosis process happens, they get pulled apart, and you're left with what are called single chromosomes, again held together by a centromere. But really, this is one chromatid. If you look at the double chromosome over here, so this is one chromatid here and here in the blue, and then here's another chromatid in the red. And again, these should be mirror copies. That's why they're referred to as sister chromatids. They should be not mirror copies, excuse me. They should be exact copies of each other. They should have the exact same DNA. So make sure you're clear on that idea that single chromosome, double chromosome, they're both just generally referred to as chromosomes. Again, sometimes you hear the term double chromosome replaced with the term replicated chromosome, but the double chromosome is the one that looks like the X. The single chromosome is the one that just has 2 of these arms. And again, this draws back to our previous learning on the cell cycle. So most of the time, the cell is not going to show the chromosomes in the nucleus. So you'll obviously see them during mitosis and cytokinesis, but you won't see them here in interphase for the most part. All the DNA does get replicated in the S phase and has that DNA until uncoiled, I should say. It's not really in chromosome form until prophase. That's when the DNA coils up and you can actually see the DNA appear in the nucleus. And again, that's what S means in the cell cycle. S stands for synthesis, the making of more DNA. So each of the 46 chromosomes in a human cell gets replicated, duplicated, so that you have enough DNA for the 2 eventual new daughter cells here. I like this picture in that it gives descriptions. I don't like it that it shows the chromosomes at each stage. You shouldn't be able to see the chromosomes in G1, S, or G2. You should only see the chromosomes during mitosis, and that's it. Now, part of understanding what's going on in the S phase is also understanding how the DNA gets replicated. And this can be a bit tricky, but there was 3 original thoughts over how DNA doubles itself. How does it copy itself? There was this conservative theory, the semi-conservative theory, and what's called the dispersive theory. So up here at the top, you can see here's the original parental DNA. The conservative theory postulated that when the DNA duplicated, replicated, however you want to say it, basically you took the original strand and just made a brand new entire DNA strand by copying it. It's relatively straightforward. Called conservative because the original strand stays intact. And again, you would undergo perhaps a second replication, and again, you would copy the original so you get this, and then this original would go here, and the new copy would be here. So it's a conservative manner of replicating the DNA. The semi-conservative theory was that, okay, something's happened to the DNA and one strand of the DNA stays original and the other strand is made new. So you get half and half, and then the same thing over here. So that's why it's called semi-conservative. Half is original strand of DNA. The other side of the double helix is a newly assembled strand. And again, you would see that replication occurs second time down here where half stays original, the other half is replicated, and you get this whole new strand because this yellow strand up here comes down here and it gets its own base pairs again. The other thought was what's called the dispersive method, where the original strand gets cut or sectioned off, kind of like chopped up, and then gets intermixed somehow with new segments of DNA, even though it should all be the same. So you get parts that are original DNA from the parental strand of DNA, and then parts that are newly assembled on both sides, and you see that process happening again. And a big part of the 20th century's question on genetics was how is DNA duplicating itself? Which of these three manners of replication is the cell typically using? As it turns out, DNA replication is kind of a tricky thing because DNA has directions to it. Not to say instructions, but it has a directional side to it. On each end of the DNA, on either side, there's what's called a 3' end or a 5' end. And I'm using the bottom strand here of this little model. On the other side, the DNA has those same labels, but going in the opposite direction. So it's what we call anti-parallel to itself. One end is running in one direction from 3' to 5', and the other one's running in the opposite direction from what we call 3' to 5'. A good analogy might be to think of like neighboring subway tracks, where you have two trains running along the same route, but they're running in opposite directions to each other. And that presents DNA with a bit of a problem when it comes to replication, because you're running in opposite directions in how you read the DNA. You have to adjust for how you replicate this thing. And typically what happens is there's some enzymes that will open up a small, I guess you would call it replication bubble. It's called a replication fork in many cases, where the DNA starts to become doubled. So you see this little gap open up. This is the DNA getting ready to be synthesized in the whole S phase of the cell cycle. But again, it has to work this way because the directions of the DNA is what we call anti-parallel to each strand. So each strand is anti-parallel to each other. They run the same way, but in opposite directions. Again, think of a train, two trains running in the opposite directions along the same path. This anti-parallel nature means the replication process is somewhat unique. One side, what we call the leading strand, which runs on the original DNA strand, which is up here in the lighter blue from 3' to 5', it's allowed to be copied continuously. The enzymes that will copy the DNA will match up base pairs, A's to T's, C's to G's, as you guys have learned, and it'll just continuously run as another enzyme is opening up the DNA and making this replication fork unzip further and further. But the other side can't run very long because you have to keep waiting for this fork to open up more and more and expose more of the DNA to be replicated on this side that's going from 3' to 5'. And so it's done in these small sections, which are called Okazaki fragments. Okazaki was one of the gentlemen who helped discover how this works. This is what's called the lagging strand. So the one that's running continuously, the leading strand, the one that's running discontinuously, the lagging strand. It's created by keeping one side original and the other side is being done in these several small fragments, these Okazaki fragments. While the other strand, this is the original strand again in the light blue, but you can continually add more as the enzymes just keep unzipping more and more and more. And there are many enzymes doing this. One of the enzymes is unzipping these things, but the big enzyme to know is this guy, DNA polymerase. This is the one that will actually go through and match the A's and T's and G's and C's, both on the continuous side and on the discontinuous side, but on the bottom side, the lagging strand, it has to do it in those small Okazaki fragments. And I like this picture because you can see here the replication bubble right here, where this side is going continuously along the top, and this side here, the bottom right, is going continuously as well. But the other sides are being done in small Okazaki fragments, small little sections as the DNA unwinds. So section, 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 section. And again, there's many enzymes that are actually helping in this process. There's little primers that got to be put on. and there's like splicing that has to be done to join the Okazaki fragments together. But what you can see is that the DNA is being duplicated in a semi-conservative manner. So one side is staying original here, and then the new bases are adding to duplicate the DNA. So one side is new. So one side's original, one side's new, and then up here, the top side is original, and the bottom side is new. It's just that one side is being continuously added, the other side is being added in these small Okazaki fragments. So semi-conservative means that for DNA, as I say, one strand is going to be entirely inherited from the prior cell, but the new strand, the mirroring, the opposing strand of that double helix is entirely going to be new. And that when you do it, this should be the exact same way. And this was a while in coming to figure out that DNA is replicated in this manner, but this is how all DNA gets replicated, in this semi-conservative fashion, with one side being new and one side being original. Now, there's another term that comes up that's easily confused with sister chromatids, and it's this term. It's called homologous chromosomes. These are a different thing entirely, though, from sister chromatids. Consider that in any organism that reproduces sexually, the offspring will get a single chromosome for each parent for most traits. In other words, you get two sets of instructions for just about every trait you have. One inherited from your maternal side, your mother, one inherited from your father's side, your paternal side. So both parents end up contributing to the genetic makeup of any offspring in a sexually reproducing species. And so I'm going to very much simplify this. You might get two types of chromosomes that code for the same thing. It's just they might have different versions of the genes, different what we call alleles. So again, I'm greatly simplifying this. Let's suppose H here is a gene located on this particular arm of the chromosome, both here and here, one from your mother, one from your father, that somehow influences height in some way. Maybe there's another gene that influences metabolism, so we'll say it influences weight. Again, this is oversimplifying things. And a third set of genes that influence, say, hair color. Now, just because they influence something like, say, height or weight or hair color doesn't mean they're exactly the same. So you could inherit, for instance, a gene from your mother that says code for light hair color and a gene from your father that says code for dark hair color. Again, these would be different alleles of the same genes. And this pairing here, because they are coding for the same traits, are called homologous chromosomes. Again, inherited from two different parents. Both of these go to the offspring, but again, these are not sister chromatids. Remember, sister chromatids only show up on double chromosomes, on replicated chromosomes. These are on the screen here, what are called homologous chromosomes, where you get one set of instructions from one parent and another set of instructions for another parent, but they're coding for the same thing, even if they don't have the same exact alleles for each gene. Now, a quick side notion on this, there can be mistakes in the copying process. And there can be environmental influences like toxins, radiation, things like that can cause changes to the DNA over time. These changes are what we call mutations. And I'm sure you've heard that term before, but I just want to run through real quickly some basic what we call point mutations that occur sometimes during the replication process, the S phase of the cell cycle. Point mutations typically are a single error in the copying process at a single base in the DNA sequence. And there's three types. There's what's called substitution, where if this is your normal strand of DNA, obviously there'd be another side here. I'm not showing the other side. If this is your normal strand though, A, G, C, T, C, substitution point mutation might have you having a resulting copy that looks like this. Notice what's going on. We have substituted in the guanine for the cytosine, hence the name substitution. And insertion, again, if you have this DNA A, G, C, T, C, and again, there should be another strand here, I'm just not showing it. An insertion point mutation might be something like this, where a nucleotide is inserted where there wasn't one before, between the C and the T. Between the C and the T here, there was nothing, but we've now inserted an adenine. So we have a substitution and an insertion, And the final type of point mutation to be aware of is what's called a deletion. The nice things about these mutations is once you know their names, it's kind of self-explanatory over what's happening. If this was our normal strand, again, you'd have two sides. A deletion might, in the copying process, might result in a nucleotide being missing in the new copy. So again, all three of these are point mutations. And what you got to remember is that point mutations may or may not impact the genetic code. They may or may not impact the protein that's being coded for. It depends on where the nucleotide is in the codon. Remember, the genetic code with all the proteins has some redundancies built in. But if it's in the wrong spot, you can end up coding for the wrong amino acid, and then maybe the protein doesn't form the correct shape that it should, and maybe it stops working. So whether a mutation is in any way harmful or if it's just neutral, which are the more likely cases, it's very rare for it to be beneficial, but whether it's harmful or neutral, that is to say whether it hurts the organism's chance of survival or whether it just doesn't really affect it at all, those are really hard to tell unless you know the entire nature of the enzyme, or excuse me, the protein that's being coded for, and exactly what's been changed. So again, you don't see chromosomes most of the time in the cell until just before the cell is about to divide. Typically, so far, what you guys have learned is that this is during mitosis only, but we'll get into another version of this of cell division in just a moment. Remember though, mitosis is how your body grows, how your body repairs and heals itself. And this is how your body also, in a sense, builds more mass. You obviously take in nutrients to gain the mass, but then your body puts that matter to work to build you as you grow up. And again, this is part of the typical cell cycle. You can't look at pictures of the cell cycle without seeing mitosis on there. And again, remember, mitosis is composed of prophase, metaphase, anaphase, telophase. And the bigger parts of the cell cycle are interphase, and then there's this kind of stage called cytokinesis where the cell's membrane is dividing and the organelles are being split up. So really the cell cycle is composed of just three stages: interphase, mitosis, cytokinesis, with mitosis being composed of prophase, metaphase, anaphase, telophase. Remember the acronym for, not the acronym, the mnemonic for remembering the order of all this. I prefer milk and tea cold is a good way to remember the order of the cell cycle, including interphase and cytokinesis, even though those are not part of mitosis. So mitosis is used for growth, from going from an infant to a full adult organism. This is true for not just you, but from things like trees as well, going from a seedling to a giant tree in the forest. It's also how your body repairs itself. So you get an injury of any type, and assuming it's not entirely, going to kill you, the repair process is going to involve mitosis, where certain cells are allowed to die off, but other cells nearby actually begin to duplicate themselves and fill in and try to repair as best they can the injury. So with mitosis, you have one parent cell that's diploid, 2N, and you end up with two daughter cells that each are diploid, 2N as well. That's to say, in humans, one parent cell would have all 46 chromosomes. Then after mitosis, both daughter cells would have all 46 chromosomes because the DNA has been replicated, again, in that semi-conservative manner. So again, here are the single chromosomes. They get replicated. They divide during mitosis, and you end up with two daughter cells, each of which has two chromosomes. So two chromosomes here, two chromosomes here, two chromosomes here, be in mitosis. And again, used for growth or repair in the human body. I showed you this slide before, but it's just basically the same thing. In this case, we have 4 chromosomes. Again, the typical number for human cells is 46 chromosomes. That's, again, why the DNA has to be compacted into these things, so it's easier to move around. And again, these are the centrioles here pulling the DNA apart. Don't confuse that with the centromere, the little center part of the actual chromosome. And again, each of these new daughter cells after cytokinesis will end up with 4 chromosomes here and here, just like the original parent had 4 chromosomes. However, your cells can divide in a different way and for a different reason. Another type of cell division is called meiosis, and this is how your body makes gametes. Gametes being sex cells like sperm cells or egg cells. And with meiosis, the parent cell is diploid, having all 46 chromosomes, but at the end of the process, you're actually not going to end up with 2 daughter cells, but 4. You could call them daughter cells, but the typical word is gametes. And these would be either 4 sperm cells or 1 egg in what are called 3 polar bodies, depending on your gender. And all of these cells, they won't have all 46 chromosomes. They're only going to have half. They're going to be haploid. So in the case of humans, if you have 46 chromosomes up here, this would only have 23 chromosomes. So again, make sure you're clear on this term diploid and haploid. Diploid is usually signified by this 2N. The N can be capitalized sometimes. In humans, this means 46 chromosomes. Haploid means 23 chromosomes in human, or half the number of DNA. So in any typical organism, if you have an organism that has, 80 chromosomes for its normal diploid cells, its gametes, its sperm cell or egg cells will have half, only 40 chromosomes. So it turns out that, yes, some of your cells don't have all the same number of chromosomes, specifically your gametes don't. And what this allows to happen is for creatures to reproduce sexually, because again, you take one sperm that has 23 chromosomes and one egg that has 23 chromosomes, and then you can get a next generation that has the full complement of 46 chromosomes. So it turns out meiosis is how your body reduces the amount of DNA so you don't get what we call DNA overload. Consider that if you didn't reduce the amount of DNA, if you just stayed using gametes at 46 chromosomes, if you had 46 chromosomes in a sperm cell and 46 chromosomes in an egg cell, you'd end up with 92 chromosomes in the next generation, and then the generation after that would be 184 chromosomes, and you end up with this thing called DNA overload. So meiosis is used specifically to produce the sperm and egg cells to cut the number of chromosomes in half so that the number of DNA overall doesn't change from generation to generation. Again, every sexually reproducing species has a manner in which that occurs. The numbers aren't always the same, but again, they undergo meiosis. They cut their DNA numbers, chromosome numbers in half so that the following generation gets the exact same amount of DNA as the original parents. Again, it's slightly different from mitosis. There's some slight variations in the steps. There's more steps to do meiosis, but it's typically only reserved in humans for production of sperm and egg cells. Mitosis is by far the more common process because it's used for growth and repair, a much more frequent occurrence. Now, meiosis, as I said, has different stages. It actually also has 2 major setups. So here you can see a basic setup for meiosis. You have a single parent cell. In this case, the diploid number, the 2N number, is 2. There's 2 chromosomes here. And at the end, you get 1, 2, 3, 4 chromosomes, each of which has only 1 chromosome, 1 single chromosome apiece. And the stages, again, are slightly different. There's a part 1 called Meiosis I, which takes you from here to about here. And it's composed of what's called prophase I, metaphase I, anaphase I, telophase I, and cytokinesis. Notice the order is still the same as mitosis. But then there's this second division, which takes you from here to here, composed of prophase II, metaphase II, anaphase II, and telophase II, and another cytokinesis. So there's this actual double cellular division going on for all of meiosis, and divided into what we call meiosis I and meiosis II. So again, if you can remember, I prefer milk and tea cold. If you can remember the prefer milk and tea cold, the milk and tea cold part, and just remember to add a 1 and 2 behind it, you know the steps for meiosis then in order without too much trouble. I like this picture. It's good. It's not showing you everything I want to talk about with meiosis, but again, you start with 2 chromosomes up here in the parent cell. They're diploid here, but you end up with 4 daughter cells that are each haploid, having only 1 chromosome. The DNA gets replicated, again, from single chromosomes, to double chromosomes, again, in a semi-conservative manner. The first meiosis step actually pulls these things apart. So you cut the amount of DNA in half here, and then meiosis II, the tetrads here, these X's, will be pulled apart again so that each new gamete gets only half the original amount of DNA as the original parents. So you end up with, again, 4 gamete cells that are haploid from 1 parent cell that's diploid, and that's in contrast to mitosis. So another unique feature about meiosis that makes it different from mitosis is that when you have these X's, and there should be centromeres here holding these 2 things as X's together, so close to each other, you can have them form what's called a synapsis. where the two X's are so close to each other, parts of their arms, their Q arms or their P arms, will actually exchange from one side to another. So you get this unique variant right here that's never existed before, and also right here, where you get this intermixing of DNA that doesn't match anything that's existed in any of the other gametes. This location is called the chiasma. I know it's CH, but it's CHI, so it's pronounced chiasma. And you see this typically with sections of arms. It can be like sections in the intermix part, but it's typically sections at the end. And this is probably the best image for copying this down that I would recommend. You can see again, here's the genes. If you consider these letters A, B, C, D, each as genes, when you have homologous chromosomes and one arm gets too close to another, this chiasma will form and you get whole sections that cross over. So you can get an intermix of genes. Now there are a video and an animation I think I'll try and show you in class that shows this process. Just know that this crossing over event does not occur in mitosis, and it's a way to get unique genetic combinations in the meiosis process that wouldn't occur any other way. It creates genetic variability without actually doing any mutations. So the DNA has not been changed itself, it's just been moved from one chromosome to the other so that the gamete that ends up with this DNA is different from the gamete that ends up with this DNA, which is different from this one, which is different from this one, because again, you're going to get 4 gametes out of the process of meiosis. So this actually shows a little electromagnetic image of the chiasma forming here between 2 chromosomes. So you can see here the synapses, the pair of homologs here forming the chiasma, and then you get this crossing over effect here at the ends of the chromatids, specifically where the telomeres are. So the synapses forms them, they have the actual crossing over event, and then again, meiosis results in the reduction of chromosome numbers. These are all unique aspects of meiosis only. Mitosis doesn't do any of this stuff. And again, these resulting cells, these 4 haploid cells, might become 4 sperm cells, or they might become a single egg cell, and what are called 3 polar bodies if the organism is female in that case. They're still called daughter cells technically, that never changes, but again, we've reduced the number of chromosomes from 2 up here, the red and the blue, down to just 1, but notice each of the daughter cells has a unique genetic combination now. So, excuse me, this is just one more image showing meiosis. Again, meiosis I up here, where the cell is cutting the amount of DNA in half after a bit of crossing over. You can see the color variations here. So you end up with these 2 cells. And then meiosis II, where the double chromosomes are pulled apart. And again, you end up with 4 cells, each of which has only half the DNA. So this has 2 chromosomes, whereas the original parent had 4. Here's an actual electro image under a microscope of this process. Again, meiosis I going across, and then meiosis II. Again, you can see 4 nuclei starting to form there. So again, meiosis is used for producing gametes. It can only be done in sexually reproducing creatures, and it's in contrast to mitosis, which is used for growth and repair. That whole system of crossing over doesn't occur in mitosis at all. It only occurs in meiosis, but it's a unique way of getting genetic variability without mutations occurring. So it creates genetic variability without making the DNA what I would call unstable or unpredictable.